Welcome to The Mixer. I'm your host, Andy Marshall. I'm here with my friend, Juan Carlos Lopez.
Hello, Andy. How are you doing?
So what's the big idea behind the podcast?
Well, Andy, during our tenure at Nature as editor of Nature Biotech, Nature Medicine, we had the privilege of interacting with a lot of great scientists who have made important contributions to drug discovery, to translational science. And we thought it would be a good idea to sit down with them, share a cocktail, and have an interesting conversation that we can share with our listeners.
Great. So who's our first guest?
Today we have Kirkpatrick, an old friend, chief editor of Nature Reviews Drug Discovery. He's fresh from a conference on RNA therapeutics, and he's going to give us the lowdown on that subject.
Great, let's get stuck in.
Let's do it.
So Pete, such a pleasure to have you here with us. And thanks for coming all the way down to New York from the dark woods of Worcester, Massachusetts. So you've just spent, a couple of days up at this conference?
Yeah, it's been a couple of days. Thanks very much for the invitation. It's a pleasure to be here on what looks like a day in London.
Yeah, for our listener, it's kind of pouring down cats and dogs outside. So this meeting essentially was put on by Nature with a couple of companies, wasn't it?
Yeah, so Nature and Alnylam, Moderna, and it was hosted at Worcester Polytechnic Institute, have a quite close relationship with the University of Massachusetts. And I think they had a lot of the kind of pioneers of actually all kinds of RNA platforms. We had people from siRNA. There was a lot of people from Alnylam, from like John Marinori, who's recently retired as their CEO. And there was a strong presence from Moderna as well. And people also working on all types and flavors of base editors, CRISPR, and just a huge diversity of platforms. But also, I guess, this kind of common thread amongst the platforms, which is really how to deliver whatever it is you're trying to where it needs to be. And particularly if that turns out to be beyond the liver, then, that was the kind of key topic of conversation.
Interesting. How much of the conference do you think was, I'm only asking this because, Moderna was one of the organizers. How much of it was about kind of mRNA vaccines and therapeutics?
I would, surprisingly little was about mRNA vaccines, and I think perhaps reflecting that there's been so much debate in the last and discussion in the past couple of years that if you didn't know about mRNA vaccines now, you've probably been living under a rock. But there was certainly some discussion on I guess the next opportunities for mRNA, in various areas where you might be able to apply them as therapies. But actually, just coming to that, one thing that was kind of fascinating. So one of the speakers there was Kathleen Carrico. It was perfect timing. When she agreed to make a presentation there, she hadn't won the Nobel Prize, but obviously quite recently she has. Still very graciously, still came to the meeting, even though it sounded like she had an absolutely incredibly busy schedule doing all of the things she needed to do for the Nobel people. But she was talking in her presentation, which was really like it was like 50 years, a fascinating tour of like research on mRNA. But one of the things she mentioned, just to your topic, that discussion there, was that she was just before she joined BioNTech in 2013, after a long string of academic things which didn't work out for one reason or other, that people are not recognizing the importance of what she was doing. She was one of the organizers of this meeting that was the first meeting on mRNA in health. And actually, at that point in time, 2013, vaccines didn't feature. So it's just it's I guess one of those things that just kind of shows you never really know exactly when things are going to be important and what and what directions things will go in. That at that time, everybody was thinking about opportunities for mRNA therapies in various different directions. So it was a discussion topic. I think most of the things here, I think it's trying to work out, how to optimize various aspects of the mRNA platform. It's fortuitous in some ways that this kind of pseudouridine that modification that was discovered by Katalin Karikó and Drew Weissman. It's a little bit immunogenic. It's still, so it still leaves this, kind of immunogenicity, but which in the context of a vaccine is desirable. But when it comes to, mRNA therapy, you're really looking to, be immunologically silent. So there's, I guess, multiple dimensions that people are kind of working on here, to optimize mRNAs, partly to remain sort of hidden to the immune system, but also to deal with the challenges of mRNA stability, which is going to be very important for the getting the level of protein expression that you need to make a therapy. There were some very interesting presentations that kind of talked about this kind of interplay between mRNA structure and mRNA codon optimization, and how these two things kind of it's really a multidimensional problem. You're trying to kind of control lots of different things. You want to optimize your protein expression, but also you need your mRNA to be stable in a vial as well, and to be something you can manufacture. This all of these things and these kind of complex parameters are kind of there's an interlink between the mRNA structure, and particularly mRNA has a tendency to sort of form secondary and tertiary structures, and also the kind of codon choice, which is something that people have used to try and optimize protein expression. So it came up in multiple different areas, and people were very interested, obviously, in like what's the next pseudouridine. I mean, I think it seems that most of the mRNA therapeutics at the moment are using a slight modification. I think it's a 1-methyl pseudouridine. Somebody specifically asked Katalin Karikó, what's the next pseudouridine, and her answer to that question was, it's all about chemistry. But she didn't have an answer to that. If anybody's looking for the answer to that question, I think. It's work for chemists to do. There's chemistry as a theme was a really common, thread amongst a lot of the presentations across platforms. Particularly there were some very nice presentations on siRNA, and I think this is an excellent example of when you go from the first generation to the kind of current generation in the clinic, just how much chemistry has happened, in terms of the optimization to get siRNA where it is now from where it was in 2003, I guess.
One of the things that I've always found super interesting about mRNA as a therapeutic is you were mentioning the kind of secondary structure stuff, Pete. I mean, it's this enormous molecule, yeah? I mean, thousands of bases where you have the molecule kind of pairing back on itself, and you have these loops, and this all relates to kind of stability as well. So this kind of secondary structure stuff is part of the equation, together with the different chemistries that you're using, whether it's pseudouridine or 5-methoxyuridine, whichever chemistry that they come upon. But it seems like just for our listeners' benefit, could we just kind of take a few steps back and kind of think about, what are the main differences in your minds that having an mRNA therapeutic poses compared with a vaccine? Because they are really different challenges, yeah?
That's an excellent question. Yeah, and I mean, and I think there are commonalities and then there are differences. Yeah, so I think with the vaccines, as I kind of alluded to a little while ago, some level of immunogenicity is acceptable, but you still, whereas in the case of the therapeutics, you're really trying to be immunologically silent. But I guess in both cases, you're looking to achieve a reasonable level of protein expression. The difference, rather, I guess, really, when it comes to the mRNA therapies, depends a little bit on what your application are. So, in the case of the vaccines, the longevity of protein expression maybe doesn't have to be that high or long. Whereas, depending on what you're intending to do with your mRNA therapy, you may want to be achieving quite, a reasonable length of protein expression, and I guess one way of achieving this to some extent, is the level of stability of the mRNA construct. I think that seems to be the kind of area of focus, how to improve mRNA stability, but also how to improve the amount of protein that's being made. And because of these tightly interlinked things between the secondary and tertiary structure that the mRNAs are adopting, and also how the choice of codons interplays with this. I guess one thing I guess perhaps I didn't mention previously was that the problem with immunogenicity to some extent arises from the presence of just uridine in an RNA, which is recognized by the immune system as being potentially viral of viral origin. And that's really where the sort of pioneering replacing this is the thing that enabled the delivery enabled mRNA to delivered exogenously to actually do something constructive. But one way of getting rid of uridines, without having to make any modifications, is to change the codon choice. In some cases, you change the codons such that you're producing the same amino acid, but you're not using a uridine in the codon. And so people have been focusing on that. I think, correct me if I'm wrong, Andy, I think CureVac for a long time had been very much about codon optimization. They didn't want to use kind of modified uridines or anything like this. They wanted to stick on codon optimization as their strategy. But I think I'd seen this, not at the meeting, but relatively recently, it seems that they've opened up to doing both.
Yeah, it's going to be really interesting to see how the different chemistries work out, and which is I suspect that it's going to be a different chemistry for the mRNA therapeutics compared with mRNA vaccines. Yeah, did you get that impression?
Yeah, that definitely came. I think there's actually also some other stuff that this actually interesting that this wasn't a huge topic at the meeting, but certainly is an important one in the field. And I think just to kind of paint a picture, I guess, of the typical mRNA construct. It's got the coding sequence in the middle, but then you have these modifications. Well, you have a 5' end and a 3' end of these UTRs, and also a cap on, and this this 5' cap and and what's going on in these UTR sequences. These are also really like a heavy area of focus in terms of achieving things with therapeutic mRNAs. So. This wasn't a huge topic at the meeting, but certainly things I've seen elsewhere, and stories we've covered in the journal, I guess. Just optimizing these things are really a key tool to improving the lifespan of mRNAs, but also what you're doing with them as well. There were actually there was just one presentation that I was on this other sort of kind of fascinating thing that you might want to achieve with mRNA therapies, and so to achieve like cell type specific expression, some quite neat idea where you basically sort of exploit the idea that microRNA expression varies a lot across cells, and you can potentially can basically harness this if you design into your 3' sort of UTR in your mRNA. You can various things that interact with mRNAs. You can potentially sort of switch off your mRNA in some cells rather than others. So this, it's a little bit into the future, but this potential to harness endogenous mRNAs to achieve cell type specific expression of your mRNA therapeutics seemed like a really interesting opportunity.
Yeah, I'm quite interested about this in a slightly different way, and I guess I have I don't know if it's two questions or if it's the same question that we need to unpack. Moderna, as you know, they already have some molecules in clinical development, and one of them is pretty close to me because it's for propionic acidemia, right, which is the disorder that my son has. And they've already recruited a few kids for this phase 1-2 study. And the results that they're beginning to share are pretty positive. So that's quite interesting because this condition has affects a protein that's the protein has two different polypeptides, right? So what they have done is to have this dual construct that expresses both, so that you can treat every kid with the condition. So it's a pretty massive construct, right? It's pretty large, and so they went ahead with the current chemistry. So I'm sort of wondering. I'm wondering if the chemistry is really good enough, and what people are trying to is just tinkering with it to get a little more stability out of it, or if we are still looking for something fundamentally better than this. If companies are already going into clinical development with the current chemistry, I'm wondering if people find it sufficiently satisfactory, and it's only a question of tinkering with it.
It's an excellent question, JC. Yeah, and I think one thing, certainly from the questions in the audience, I think there were various people who are quite expert in the field, I guess, making this analogy with the history of siRNA and also antisense oligonucleotides, and just like what the first generation chemistry there looked like, and just how different what it is now looks. And I think if you can achieve more with the chemistry, and actually Kathleen Carrico and also Anastasia Kovrova, they were all very much kind of like chemistry is going to be the answer to this question. So but there's also actually there was one thing I really wanted. I thought it was a really important comment that kind of came from Melissa Moore, who's recently retired as CSO of Moderna, and I think it's a really important difference with say siRNAs and antisense oligonucleotides is that basically they were able to do a lot of modifications in those cases on the 2' hydroxyl. She made the point that actually that in the case of mRNAs, that 2' hydroxyl is really important for the fidelity of transcription. So essentially, you can't really mess around with that, or you have to be very careful messing around with it. Otherwise, you're not going to end up with what you hope you're going to end up with as far as the right protein. I think like the type of chemistry may be different. So this is maybe, I think the great thing about this sort of parallel development of antisense oligonucleotides and siRNAs, a little bit separated in time, but at the end, some of the same chemistry was taken from one platform to the other. And, there's these things where people have solved the problem and then realized that they can use it in both. But yeah, with mRNA, perhaps, this is just me sort of anticipating, I guess, is that I think there may be some novel chemistry that is needed, or, ways to, to modify things. And yeah, there's also this question, I guess, and I think that's something you said, Andy. The other difference really between, I guess, mRNAs, therapeutics, and the siRNA and ASOs. ASOs and siRNAs are short. That you can make them easily with chemical synthesis. mRNAs are gigantic. And the current sort of way that they're made is through in vitro transcription. How you do this chemical modification is just another level of challenge, I guess, when it comes to mRNAs. I'd love to know, actually, this is not something I'm super familiar with, but a couple of people make comments that, if you want to make chemical modifications to mRNA, regulatory agencies are going to be nervous if that isn't done consistently. I think you need to be able to have a very strong characterization of whatever chemical modification you're making to your mRNA if it's not something that's done throughout. It's the early days as far as mRNA chemical structure optimization beyond what's been achieved so far, I guess. Maybe you can. I'd love to know how this works, actually, because it's not something I'm super familiar with. But the actual mRNA manufacture process. I'm assuming you feed, your in vitro transcription the components that you're looking for.
Yeah, that's my understanding, Pete. But I think there's very, very little out there where people kind of talk about this stuff.
Yeah. The manufacturing side of stuff is, one person from Moderna, said this is, it's another aspect actually of the immunogenicity problem, and I hadn't really appreciated this before. But the way that mRNA therapeutics are manufactured, it's in vitro transcription, but a little bit of a contaminant of double-stranded RNA, if that gets into the body, then this is going to provoke an immune reaction. So there's actually a whole in parallel kind of optimization, of like sorting out your mRNA manufacturing process. And also in some cases, people have been working on better TCM polymerases to get to essentially result in a product that's, as free as possible of any double-stranded product. There have at least been a couple of papers put out on this, but I think, it's a little bit like the lipid nanoparticle manufacturer, like it's a bit of there's a lot of knowledge in the field that just isn't necessarily out there in the public domain, in the way that some other stuff is.
Yeah, well, I think it's somewhat similar to, if you look at the oligo field, is like a lot of the kind of proprietary chemical modifications were discovered in ISIS, which became Ionis, and then many of those chemists ended up kind of at Alnylam, and as you were saying, Pete, like they were able to use those modifications, and they worked very well. But yeah, I think this is going to be really interesting to see how it works out.
I had the opportunity to visit Moderna a few years ago. They were already beginning to think about clinical development of this RNA for propionic acidemia, and COVID was still not in the picture, right? So they had just their labs for this. And it was quite striking because when they showed us around the lab, they made the point of showing us their biggest bioreactor. And their biggest bioreactor was the size of like a large bucket. It was not one of these massive bioreactors that you see that people have for biologics. So to them, that was the power of this, that they didn't need such big bioreactors, but that they could do synthesis in a more efficient way. But this doesn't negate the question of manufacturing. I think that's kind of a challenge. Now, the other side of this,
it's probably also taken leaps, JC, with the vaccine and number of doses and the kind of attention to scale up.
Yeah.
So, yeah.
But you see, the other side of this, and Pete, you were alluding to this at the beginning, is the question of delivery, right? And certainly another way to optimize the efficacy of whatever you're delivering is to target it in such a way that it reaches the cells that you're interested in reaching in a much more efficient way, so that even if you don't have a lot of the RNA, you may still get a biological response. Was that something that they talked about at the meeting? I've heard, and this is not new. Every month somebody solves this problem of delivery, but I'm kind of wondering if there was anything there that caught your attention, or in just your reading of the literature, if there's anything that you'd find particularly exciting in that space.
Yeah, it's a great question, JC, and I think it was a real common thread at the meeting, whether what was being delivered was siRNA or mRNA or genome editing therapy, and I think one thing that's become it's really risen to the fore, I guess, as a result of the mRNA vaccines, as it's been the lipid nanoparticles. And it's almost got to the point now, like it seems a bit, I don't know, to me it seems a little bit reminiscent of the the VHS Betamax kind of thing. It's like, there are these two platforms now that people are applying, particularly, I mean, not for siRNAs, but but when it comes to the genome editing, which is the and it's AAV vectors or LNPs, and there's a lot of cool work going on with both. I'll come to the LNPs because this is relevant to the mRNA therapies. There were some presentations. There's a guy called a professor at Emory called James Dahlman, who's doing some really cool work on LNPs and the ability to kind of screen them for their ability to do in vivo editing. And and I guess the thing that was kind of coming across from his work, was this kind of barcoding approach where you have this ability to assess a large number of lipid nanoparticles and to see in a single animal where they're getting to, and to kind of optimize this in a slightly a much more not just a one by one kind of way. I think maybe you can potentially assess maybe up to 70 or so lipid nanoparticles in an animal, and this opportunity, to do lipid nanoparticle optimization in vivo at a larger scale. My impression is, from both the talks that were there and also from kind of conversations with people elsewhere, is that the LNP platform is making substantial progress. I get the impression, that certainly some people were kind of somewhat confident that this is about genome editing, but they were optimistic that that people had developed lipid nanoparticles that were capable of targeting hematopoietic stem cells in vivo, so which would be a real breakthrough for this. It certainly seems. Yeah, that there's a whole. I mean, actually, if you look at Nature Biotechnology, I think these days there's quite a few of these kind of barcoding LNP kind of things. It seems to be, it's a new hot trend in terms of, and actually, commenting on that, I think James Darman was really interesting. I think he was basically highlighting the differences between the kind of assays they would use for publications and the kind of assays you'd need for clinical translation. And his comment was really that that they're not the same thing, and there aren't that many people who are selecting their LNPs with the kind of assays that would really work to work for the ideal ones for clinical translation. Also something that I wasn't at the meeting, but I think this was from John Andrzejewicz at Pfizer. It's basically not that many people are aware that rodent models are not really very good predictors of the efficiency of LNP delivery in humans, and some people just a lot of people are working in the area not so aware of it. So, I think there's definitely this kind of issue around, finding the right assays to identify the right LNPs, but certainly the technologies. I mean, there are a few companies out there now. I mean, James Darman's one, he basically founded a company that got acquired by Beam. There's another company out there from Mike Mitchell's lab in the UPenn, Liberate Bio, that are working on the similar kind of coding thing to kind of come up with better LNPs. It sounds like there are any number of other stealth companies out there also working on cool ways to develop LNPs.
So Recode Therapeutics. So was it that was that kind of Nature Nanotech?
Yeah. So
there's so much going on.
Optimistic. Yeah, I mean, I think there was optimism. That certainly, the nature of the LNP system, and the ability to kind of optimize the various different components. If people can get the right assays and the right to optimize for clinical applications, there's the potential to address some of these delivery challenges.
So it seems to me like there is, with LNPs at least, there are some problems that we've solved. Yeah. So with the vaccines, it's intramuscular delivery, and then we know that they get to lymph nodes and they get to the spleen. So those are two kind of organs that we know we can get to with the existing LNP technology. And then, of course, everything else goes to the liver, yeah? Mm-hmm. The liver is the sink where everything else gets sucked. But then there are so many other diseases. I mean, some, obviously there's the CNS. Obviously there's muscle. There's so many muscle diseases that people are interested in. There is the kidney as well. Mm-hmm. And now you mentioned the bone, yeah? So getting some of the hematopoietic stem cells in the bone, that's an important leap, as you said. But did anybody mention any kind of work that shows all along? That's another one, yeah?
Yeah. I mean, there are a couple of things actually that I just I think, to that question. I mean, so Kevin Fitzgerald from Mount Island did a presentation. He was talking about various programs that Mount Island have. I think, they have this interesting challenge at the moment, I guess, of so many things they could try and trying to work out which ones to prioritize. But, one that was exciting was their program in Alzheimer's disease. So, this is in phase 1 at the moment, and they've demonstrated delivery of an siRNA to the CNS for this. So, and they can see that it's having an impact on APP levels. I think as far as siRNA delivery to the CNS, they've managed to achieve enough to do something impactful potentially in Alzheimer's. This is a very early stage clinical, but I think that's given the challenges that people have long pointed out about the anti-amyloid antibodies. This potential to take out, things that you want to take out, the level of, the siRNA is doing is potentially really interesting. And he also made an analogy where, with the area where, siRNAs have shown some impact clinically already, and so ATTR, and it's, their impression there is that if you can stop the production of ATTR, you do end up with kind of clearing plaques basically. So this hope that might be able to achieve the same thing in Alzheimer's was it's a very early stage thing, but it certainly sounded like at least they'd achieved the sort of step of the delivery challenge part of this problem.
And that was LNPs, was it?
Yeah, they were used. Actually, no. Sorry, this was this was a Galnac. So this is the other side. I mean, actually, so that's I really changed topic there a little bit. But yeah, because actually, siRNAs are quite interesting in that like ASOs with these two different approaches to delivery. And the first siRNA to make it to the clinic was using an LNP, patisiran. Yeah,
right.
But basically the ones that have come in since have used Galnac and have exploited the ability to get across.
But that's to the liver, yeah.
To the liver, yes. So sorry, the conjugate they're using for the I don't think he didn't disclose the conjugate.
Yeah.
But they say they've because I think you need a particular it's another aspect of the problem, I guess. You need the right conjugate to get across into the right cells. So that wasn't a focus of his presentation. But I think this is an area in which obviously people are, coming up with the next ASGPR when it comes to delivery is another kind of key thing. It's worked out so well for Galnac just because of the high expression of the ASGPR on liver cells. But yeah, being able to find similar cell surface receptors that have got like high concentration in a particular place, and also I think the other aspect he was kind of focusing on was that you're not disrupting some endogenous signaling pathway by jumping on the ASGPR to deliver your siRNAs. So finding these things that receptors are like that elsewhere is a key part of the challenge.
Yeah, I know. I mean, Moderna had a large team of people when Melissa Moore was there, kind of working on the delivery, working on different lipids, aminolipids that have different tropisms to different. I mean, it's been a very, very big kind of effort, kind of industrially. So when they do find some of these, like there's been a few papers kind of talking about these kind of squalene aminolipids that have some pretty interesting tropisms for different tissues beyond the liver. To me, this is the kind of the real frontier. If any of these kind of prove to be, something that opens up a tissue, then similar to the way in which Alnylam went after the liver, and now everybody can get things to the liver. If one of these other organs opens up, then I think it'll be a seminal type of event.
A lot of people already agree that the mouse is or the rat or these small animals are not particularly good models for this, and yet people continue testing there. Right? In some of my interactions with the investor community, people worry that unless you've shown that this works in non-human primates, then we're not really interested. And one needs to pay attention to that, because the amount of work you see in academic institutions in terms of targeting nanoparticles to pretty much every organ, and the number of papers and abstracts that get written about this is staggering. And people are not paying enough attention to it.
It's the same issue with vectorology, with gene therapy. It's like,
the same,
the primate is not the same as a mouse. Neither of them are the same as a human. If you're talking about the CNS, and then you have like ages, like the vascular changes with age, disease tissue changes. So I mean, some people are thinking, the best way to go is to start with human. You can do a lot of work now, kind of analyzing human tissues. Single cell RNA-seq, and kind of work out which things are expressed on the surface of human vasculature in certain tissues, and then you choose your target of choice, and then you go back into your model, and you make your mouse model with that human target, and then once you've done that, then you can start to do the work, and you hope that will be informative, but it's really tricky.
So Pete, shifting gears a little bit, now that we're starting to talk about Galnacs and other kinds of molecules, One area that's of great interest to me, at least, and I'm sure that to a lot of other people, is this idea of targeting RNA with small molecules, right? So the target in this case is the mRNA. Was there any discussion of that at the meeting, or is this an area in which you've seen anything exciting that you would like to share with us?
It's a great question. Actually, there was one speaker there, Xuan He, who does some work in this general area, but I think he looked at the rest of the program and he realized that most of it was about either siRNAs or genome editing and decided to talk about something else. So this topic really didn't come up so much at the meeting. I think just a reflection of the composition of the speakers. But a couple of people, I guess, highlighted one of the key diseases where this it's proven to be possible to come in with a small molecule and target RNA therapeutically. And this was spinal muscular atrophy. And it was how interesting it was actually that basically there's an antisense oligonucleotide, nusinersen, been on the market now for a few years, which promotes the kind of exon skipping and SMN2 that's needed for this disease. And very interestingly, phenotypic screening was able to identify a small molecule that did essentially, the end result is the same. It promotes exon skipping, and I would say, it feeds into a very interesting kind of question. A little bit what we were talking about with assays, I guess, is that if you can come up with good phenotypic screening assays, that have meaningful linkage between, the screen and the clinical translation, as was the case in SMA and the small molecule screens that were used, that was with Roche, I guess, which, we're working on this. There's really a huge potential still for small molecules targeting RNA to achieve interesting things. So it wasn't a topic that came up at the meeting, but I think we've published an article relatively recently from Matt Disney, who's been doing work in this area for a long time, and has also, been identifying all sorts of interesting strategies to more rationally target RNA structure with small molecules. And, I think there's a huge amount of potential in the area. But it's not something that came up recently.
Do you think, Pete, that there's a lot of skepticism about it as well, yeah? Because RNA structures are really tricky to drug.
Yeah. I think this is it's one where the tools for characterizing what are going on is really, I guess, kind of pivotal to achieving something constructive. But also, I guess, in the case of the drug for SMA, it feeds into something which is, completely, it's quite peripheral, I guess, to the topic of this meeting, but actually very important for therapeutics in general. We published an article from a guy called Jack Scannell recently, and it just, one thing that kind of really, it was emphasizing so much in this is the predictivity of your screening tools, wherever they are in the process is such a kind of fundamental thing to the ultimate success of your project. But it's just amazing how little sometimes people question, the predictability of the stuff they're working on. They carry on using the same assays the field has used for a long time, and and unsurprisingly, at the end, sometimes things don't work yet again. And it was just calling for, I guess this article was very much calling for more investment in better models at whatever stage you're using. I just thought I'd mention it because it's a topic that actually I think, it cuts across a lot of different things, and perhaps just doesn't get the traction it needs from people awarding grants or also, funders of all types.
The two that always come to my mind was it's Evrysdi, that was the SMA drug from PTC, and then there was Atulurin? That was
from PTC.
Duchenne,
Yeah.
But they weren't super convincing.
No. I think particularly the Atulurin one. It's still, I think it's approved in Europe and still not yet approved in anywhere else. Yeah, certainly the FDA. I don't think we're interested. It really speaks to the challenge of achieving something meaningful clinically. Some of these things, I guess, with so many different platforms out there now, the question is, in which diseases would you choose to go after them with a small molecule targeting RNA when you can see the emergence of all of these alternatives that are really heading for the root cause in a much more direct way?
There's another wrinkle on that, isn't there? Which kind of may also speak to JC's question, which is this idea of using a drug to inhibit a RNA modifying enzyme or protein. So I know Storm is kind of one of the pioneer Storm Therapeutics in the UK with metal free, which depletes M6A. They've been looking at that in cancer, and there seems, correct me if I'm wrong, but it seems like there's been quite a bit of activity there as well. Yeah.
Yeah, I agree. I mean, again, I think it's a fascinating field. This whole aspect, wasn't one of the common threads at the meeting. But I would say, there was a whole bunch of platforms. I mean, I don't know if it's helpful to talk about this, but I think the other real strong theme of the meeting was the various different flavors of genome editors. And this is it's a really exciting year for us, I guess. We've seen the submission of the first therapeutic based on CRISPR to the FDA earlier this year. I guess there's a possibility that this year may see the approval, I guess, of the first kind of CRISPR-based therapy for these I'm going to just stick with sickle cell disease and beta thalassemia rather than trying to say hemoglobinopathies again. If this happens, I mean, the speed, from the kind of CRISPR papers in 2012 to a potential drug on a new platform just over a decade later is incredible. I think saying that, the thing is that what was also very interesting at the meeting is just how quickly the field has moved on, from the kind of first generation, CRISPR strategies to, the various different flavors, which I'm sure you're very familiar with in Nature Biotech, Andy, all types of base editors and, these days prime editors as well, and these kind of next generation platforms and their ability to, to take things to the next level. And particularly the first application, of the genome editing is to do the thing, I guess, that, CRISPR's good at, which is disrupting the function of a gene, and in this case, it's quite a neat idea of knocking out, the function of BCL11A, and in that way, essentially what you're doing is leading to the reactivation of hemoglobin, the fetal hemoglobin, and this is what's enabling the treatment of sickle cell disease and beta thalassemia. But what's really exciting, I think, and what was really like a common theme amongst several talks at the meeting, was the huge potential of the particularly base editors, because these are the ones that are entering the clinic at the moment. There were some really nice stories there to fix particular diseases with a known root cause in a much more precise way. David Liu gave an absolutely fantastic presentation. He's really been pioneered so much in the field of base editing and prime editing, and just to give he talked about various different generations of these. He gave a really it's quite recent paper in the New England Journal of Medicine talking about this trial. It's been run in the UK for quite a rare cancer, T-cell leukemia, and essentially he just presented a kind of case study that I think was part of this NAGM paper, of a young girl basically who really just exhausted all existing options for this T-cell leukemia, and she as part of this trial received a base edited CAR T that had 3 different modifications made to it, and it was all it was so cool to be able to do these 3 things in a very precise way with a base editor, and as a result, I mean, essentially, she was basically terminal, and now she's back at school. I mean, it's really it's one patient, but it's completely transformed her life in this case. So and I think it illustrates the potential of this base editing approach to do things that just haven't previously been possible. There was also some the other area where base editing has been taken into the clinic recently. The company Verve, who've been very ambitiously looking to to fix
cardiovascular, isn't it?
Cardiovascular disease. You think a lot of the time when it comes to new platforms, people go for things like cancer because the risk benefit it means you can. It's a lot more. There's a lot more of a window than in the case of something like cardiovascular disease, some of the time at least. But there was some really nice presentation of almost following in the path of the statins many years ago. But this this familiar hypercholesterolemia story where there was in this case they'd identified some patients with some really severe form of this disease, which I think there was an 11-year-old girl who'd had 3 heart bypass operations at the age of 11, and it's like they've administered a base editor now, and I think, as I understand it, the first clinical data from Verve's trial should be being reported later this year. So it'd be if this works, I think this is going to be very exciting for the field of base editing. I'd come back to David Liu because he's just been such a powerhouse of cool innovation. But he was actually primarily talking about prime editing, and I think base editing, is super cool. But if you can make prime editing work, it's a little bit further behind. But I think people were kind of comparing. If you think of like CRISPR as a pair of scissors, prime editing is basically like having a word processor. You can do so much with it, and this potential to just completely transform the way that some diseases are treated. There was a really interesting kind of concept. At the moment, cystic fibrosis is a rare disease where Vertex as a company have done an amazing job, kind of almost going in mutation by mutation in some cases, coming up with a pool of drugs that now I think you can treat the majority of the pool of people who have cystic fibrosis. But just imagine you could go in and the kind of coding region of the gene that's got most of the mutations in and just replace that with a functional CFTR, and this is the kind of thing that may be feasible if you can get prime editing to work.
Yeah, it's very far in the future, though.
It's far, yeah. It seems very far in the future. Yeah, this was the talking to the people at the meeting. It seems it's far into the future, but maybe not as far as it has been in the past with analogous platforms. I think if you think about the pace of innovation. Yeah, an antisense or siRNA, I guess. I'll come to it. There was a really nice presentation from John Marinori about the history of Alnylam, but that's sort of it took about sort of 16 years or so maybe to get from the formation of the company to the first drug on the market. And antisense oligonucleotides. Yeah, it's actually.
Considerably longer.
Considerably longer. I suppose actually the first one made it in 1998, which is not, it's probably like 20 years after the formation. But for the platform to really kind of, to reach the point where you say, this is like, it's relatively mature, it was longer. And, now you look at CRISPR and you see the pace between pioneering studies and, the potential approval of a drug is getting to be more than a decade. So, I'm sorry, right? Just a little bit more than a decade. And, prime editing is futuristic, but perhaps I think the pace of innovation and it's maybe not so far into the future as has been in the past with innovative platforms. If some of particularly the delivery challenge can be ironed out, because what he was talking about was something really very cool. He's basically coupled the kind of prime editing technology with something else he'd invented in his lab many years ago, which is this phage directed evolution sort of strategy called PACE. And he was using PACE to optimize components of the prime editor. This ability to kind of harness directed evolution to improve the quality of your prime editors. Is something that might lead to a much more, rapid development of the prime editors with desirable properties than might have been previously possible.
I certainly agree that it won't be that long before some of these therapeutics reach the market. I think that there are a few challenges that have to be sorted out, such as, again, manufacturing, some of the aspects in regulatory science and other things that are not necessarily related to the innovation itself. But I think that that's an important area to look into.
I agree, Jay.
Pete, this has been a very thoughtful conversation, as we knew it would be. Right. I mean, we're already coming to the end of the recording. I wonder how many people are still listening. But one last question, Pete, before we let you go. So as you know, our podcast is called The Mixer. So our question to you is, what is your go-to drink?
At the moment, it's an Espresso Martini. I find this, particularly when traveling, it's what I need in the evening.
That's good for jet lag.
Exactly.
I have never been into espresso martinis, but we can certainly make one. Somebody told me about a secret to get more foam, and the secret to get more foam when you prepare it is apparently to use larger pieces of ice. That's something I didn't know. I haven't tried it because I don't make this drink too often at home, but it's something for a future episode of the mixer. And if you're game for an espresso martini,
so there you go, JC. Seminal insights, as always.
That's what I'm going to take home, definitely.
All right, Pete. Thanks so much for your talk. It was really great talking with you and really informative.
Thanks so much for the invitation. It's really been a pleasure.
Well, on that note, I think it's time to call it a day. I must confess, I'm not really partial to the espresso martini, but for those of you who like an espresso martini, we have the perfect recipe down in the description below. Anything you want to add, Andy?
No, this was great. Thanks a lot, everybody, for listening. Until next time.
Cheers.
Cheers.